Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at anaerobic respiration, which is part of your Module 5 respiration topic. So let's just take a step back though. Before we can understand anaerobic respiration, we have to remind ourselves about aerobic respiration. So when oxygen is present, what will happen is aerobic respiration will occur, and that's because oxygen is the final electron acceptor of the electron transport chain, the ETC, in oxidative phosphorylation. So when we're at the cristae and oxidative phosphorylation is occurring, those electrons can move through the electron transport chain because oxygen is that final electron acceptor. Well, if the oxygen concentration is too low, the flow of electrons through that ETC will obviously be decreased to the point that it could actually stop. Now, if the electrons can't flow, if the electrons aren't moving through the electron transport chain because there's no oxygen to be that final electron acceptor, what that's going to mean is that our coenzymes will not be able to be regenerated. So we have reduced NAD comes from glycolysis, reduced NAD comes from the Link reaction, reduced NAD and reduced FAD come from the Krebs cycle. So if these coenzymes are not able to be regenerated back to their oxidized state, then glycolysis, Link's reaction, and the Krebs cycle will actually stop. So let's look at what happens then in yeast cells. So anaerobic respiration in yeast cells is actually called alcoholic fermentation. Well, glycolysis can still occur because glycolysis is not needing the oxygen for the process to occur. So glucose being broken down into pyruvate through dehydrogenation, the removal of hydrogens from the molecule, and substrate level phosphorylation, giving us a net of 2 ATP. So through the process of glycolysis, ATP is being produced with a net of 2 ATP molecules. Well, when there is no oxygen present, for anaerobic respiration to continue, what happens is the pyruvate needs to be decarboxylated. So for it to be decarboxylated, what needs to happen is carbon dioxide needs to be removed and the pyruvate is converted into ethanol. Well, all that we've done right now is we have removed carbon dioxide, we've got an intermediate molecule. We're still not able to produce any more ATP molecules. So what we need to do is we need to convert our ethanol into ethanol, and we do that through the process of dehydrogenation. So we are removing the hydrogens and electrons from the coenzyme NAD so that we are still able to yield our 2 ATP molecules. But what can happen is we are regenerating NAD so that NAD can go back and glycolysis can continue. So more ATP molecules can actually be produced. So let's just look at that step by step then. So glycolysis has occurred, but the first thing we're going to do in alcoholic fermentation is where the pyruvate is going to be converted into ethanol by the removal of a carbon dioxide molecule. This is catalyzed by the enzyme pyruvate decarboxylase. Decarboxylase A's, it's an enzyme, it's a decarboxylase, so it is necessary for the removal of the carbon dioxide from the pyruvate molecule. So the name of the enzyme is giving away what it actually does. Now, once we have decarboxylated, the next thing that's going to happen is this dehydrogenation. The ethanol is going to accept the hydrogens from the reduced NAD to be converted into ethanol. We have regenerated the NAD. The NAD can therefore go off and be used in glycolysis again to keep the yield of 2 ATP molecules. So the coenzyme being regenerated is going to allow for that glycolysis to continue. Now the problem we have with yeast cells is that ethanol is actually a toxic waste product. And if the ethanol concentration gets above 15 degrees C, it will actually kill the cells. And that is how we use alcoholic fermentation in the production of alcohol. So we'll look at that when we look at module 6 and the biotechnology topic area. But let's look at what happens in mammals. So alcohol fermentation is the anaerobic respiration in yeast cells. We're now going to look at mammals, and we don't produce ethanol. Instead, we produce lactate. We do what's known as lactate fermentation. So we've got the same process that glycolysis is going to occur in the cytoplasm. The glycolysis will produce our pyruvate molecule. We end up with our 2 ATP molecules at the end of glycolysis. Well, if there is not enough oxygen present, what then happens is we still need a molecule to accept the hydrogens. And this is where pyruvate is going to be that molecule that's going to accept the hydrogens. So that is through the process of lactate dehydrogenase, an enzyme going to catalyze the reaction of the pyruvate being converted into lactate. Now, that is so that we can regenerate that NAD. And that regeneration of the NAD means glycolysis can continue. We're yielding 2 ATP molecules with every glycolysis reaction occurring. So it's important that we remember that anaerobic respiration, this lactate fermentation, only produces 2 ATP molecules, and that can only happen with this regeneration of NAD. So let's just look at that again through this process. We have the pyruvate is going to accept the hydrogens and be converted to lactate. Therefore, we have dehydrogenated the NAD, the coenzyme. We've dehydrogenated that through the process of a dehydrogenase. Lactate dehydrogenase is the enzyme that's going to catalyze that reaction. Now, coenzyme NAD is regenerated. The coenzyme NAD can now go back and glycolysis can continue. Now, the key thing about mammals and the production of lactate is that lactate actually needs to be broken down in the presence of oxygen, and that occurs at the liver. So the lactate is actually transported to the liver, and what happens at the liver is that lactate is converted back into pyruvate and then converted to glucose, and that's through a process called gluconeogenesis. Because we are converting glucose from a pyruvate and from a lactate molecule, we are producing glucose from another substance. So that happens at the liver, and that happens when there is enough oxygen present. And that's why in mammals we build up what's known as a oxygen debt, the oxygen that is required to convert that lactate back into the glucose molecule. So mammals are able to sustain a long period of time where they are anaerobically respiring, but what they will do is they will build up an oxygen debt that needs to be repaid. So different organisms actually are adapted to environments of where that oxygen is present in the environment or not. So for some organisms, for example, mammals that live in the water, they are able to sustain periods of time where they can go without oxygen, but what they're doing is they're building up this oxygen debt. They then need to come and replenish that oxygen and ventilate their system by removing the carbon dioxide and by bringing the new oxygen in. So it's really important that you understand that the liver is a vital organ in the fact that what the organ does is it will detoxify. So lactate is a toxic toxic substance in mammals, it can be detoxified at the liver by converting it back into pyruvate and then glucose. What you also have is this ventilation, which brings in our topic 3 module, where we need that oxygen to be brought into the body and carbon dioxide to be removed, because that will affect our pH if we do not maintain a balance in our bloodstream. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you haven't already done so, do check out my revision platform, www.aiqchat.com.